Hi, my name is Cassian Yee. I'm a professor in the Department of Melanoma Medical Oncology and Immunology. I'm also director of the Solid Tumor Cell Therapy Program at MD Anderson. I look forward to meeting as many of you as possible, but unfortunately, um, I can't make it. So this will have to do. I think if I'm going to summarize the sort of research MO in our lab, it's exactly this. And what I mean by that is that what you read, what you hear presented, um, always approach with a little bit of skepticism. Not that everyone else is making signs up, but you have to consider for yourself what the basic first principles are in any endeavor. I think that if you read enough papers and they reinforce a hypothesis or your own intuition, which is accumulation of years and years of reading and introspection and more reading and discussions, and all that leads you to believe that somebody's hypothesis is real, then you should go with that instinct. But I think one of the problems, especially in the field of immunotherapy, is that there are so many shiny objects. And this does lead to a sort of lemming like, um, I guess, behavior, even among scientists. And it's not that all scientists behave like this, or you know, I can even say that sometimes I am easily distracted by something interesting or exciting that I might have read in a Twitter feed or the latest cancer cell journal or what have you. But I think that immunotherapy is especially prone to irrational overexuberance. Um, and so you need to sort of step back and understand what the first principles are. And so I didn't actually tell you what I've been doing yet in research, so I'll tell you now. What I do is study the T cell response in humans, in patients, to cancer, and sort of dissect the role that T cell based therapies have in preventing, treating, preventing recurrence. And this involves a whole lot of different disciplines, not just immunology. Um, and this is one thing actually I can thank the cancer biology program here for. I think Raghu Kaluri actually was the first to engage me in this sort of research in progress meeting um, many years ago. And from there, I sort of realized, listen, immunology is actually a relatively young field of research, and all the cancer biology, all the genetics, all the epigenetics, metabolism, and everything that goes into actually building a robust immune system can't be ignored. And in fact, we should explore this multidisciplinary approach as much as possible. And so, fortunately, I've had the good luck of meeting Jin Hu and Ye Jing and others, and working with them as much as I can, and Raghu, obviously. But also people like Kunal Rai and epigenetics, across the road over at Rice University in bioengineering, understanding the cues and responses that a normal T cell circuit is required to sort of follow in order to become activated, to rest, to sleep, to be quiescent, and then become memory, and then activated again to kill the tumor cell. So I know I'm kind of a stream of consciousness here, but let me just go back and explain to you what these first principles are. The first principle is that a T cell has undergone many generations, centuries, years, millions of years of evolution to achieve a very tight control over what the T cell receptor sees, what it ignores, when it becomes activated, and then engages a productive response, which usually means release of cytokines or perforin enzymes and mechanisms to kill its target. Kills its target through T cell recognition as a first step of the peptide MHC complex. Now, what you've heard about CAR T cell therapy and TIL and TCRT engineered therapy is that you need to make a very powerful response, for example, by lymphodepleting the patient beforehand and putting these T cells in. For some types of cell-based therapies that might be important. For others, it's not clear if it's important or not. Some of it, a lot of empiricism. And so you should question: is lymphodepletion required? Um, is it because it provides a homeostatic environment of cytokines for T cells to grow and proliferate? Or is it defeating the purpose of immune therapy by um, eradicating potentially beneficial endogenous tumor reactive T cells that can lead to antigen spreading? And now you sort of just killed off all those cells with lymphodepletion. So understanding those principles and there's just reflexively saying, oh, we ought to give fludarabine and cytoxin is something we should all think carefully about when we move into the clinic. This is getting back to the T cell receptor and the whole um, cell-based therapy approach. When the T cell receptor is engaged, it does have feedback circuits that downregulate the T cell receptor. The cell enters a more anabolic state, acquires certain, I guess, transcriptional signatures and epigenetic programs that allow it to become a memory cell. And then when it needs to be activated, it becomes an activated T cell. Now, a lot of that is going to be undermined if you just replace the entire receptor machinery, just Frankensteining in something with its own activating molecule and overriding all that internal circuitry. And so I'm not saying, of course, that CAR T cell therapy has not been a good thing. In fact, quite the opposite. It's been an amazing discovery by Carl June, Michelle Sadler, and others, and it's cured many patients, saved many lives. But if you're going to go beyond that to understand what constitutes memory, what are the signals that a T cell requires, you actually need to go back to just the endogenous T cell, the one that's circulating through the body. And so that's the approach that we've taken. We very naively thought over 20 years ago that tumor reactive antigen specific T cells are already present in your peripheral blood. And in fact, when the first uh, peptide C tetramer was invented or engineered by Mark Davis and John Altman a long, long time ago, we say 25 years ago. I called up Mark, and I was barely a junior faculty member, and Mark was a professor who, pretty much sequenced the T cell receptor, and just say, "Hey, Mark, hey, why don't we just use that tetramer instead of looking at HIV epitopes that you've been doing? Let's look at melanoma-associated antigenic epitopes." And at that time, only two or three had just been discovered by Terry Boone and others. And he said, "Well, let's use tetramers for these." And he said, "Sure, why not?" I worked with Peter Lee in his lab, and lo and behold, if we screened enough cells, T cells circulating in peripheral blood, we actually found a small fraction of melanoma antigen specific T cells circulating in the patient. That was heresy back then. But from that point on, sort of took that, and maybe because I was a little adverse to molecular biology and engineering receptors, went with the sort of a more holistic approach, I suppose. But really hammered home sort of how do we isolate these endogenous tumor reactive T cells, and using some enabling technologies of tetramer guided cell sorting and then expansion, and then subsequently overlaid that when we're looking for a way to program naive human T cells into memory cells. Started using IL-21 during priming, a gamma chain receptor cytokine that actually took us 12 years to figure out, actually in the human system, exactly what it does epigenetically by activating phospho-STAT3, binding to promoter regions of CD28, and then also co-opting FOXL1 phosphorylation and so on, to drive a memory program. And the real proof of principle was when we put these T cells into patients. Um, and in a study in 2013, so this is many, many years after we'd already developed this method, and now we were treating patients with acute myeloid leukemia targeting WT1. We actually made a batch of cells that were not primed to IL-21, but was uniformly specific for this AML target, which was WT1. And we also made another batch for another group of patients using IL-21 primed WT1 specific T cells. So one set were not exposed to IL-21, the another cohort of patients that received T cells that we felt were memory programmed T cells. And then we conducted the clinical trial. The first cohort of patients who were very high risk for leukemic relapse, they received WT1 specific T cells. Unfortunately, these T cells did not persist long term. And also, very unfortunately, the leukemic cells came back, went from barely detectable to 100% within a matter of weeks. Um, and the WT1 specific T cells just disappeared, unfortunately. But when we gave the patients, um, similar group of patients with high risk leukemia, WT1 specific T cells that had been primed out 21 and had memory phenotype, you can see the T cells persist long term, and the leukemia never came back. And I think one of the lessons there, obviously, is that the autoimmune prime memory T cells were quite effective. But the second um, uh, uh, lesson there was that if you treat with low burden of disease, you could probably prevent recurrence of some cancers. And I think following on that success, we thought, well, let's go into metastatic melanoma. At that time, immune checkpoint therapies had just emerged. Um, and we felt that because the T cells that we had made with this autoimmune programming and sorting and expansion expressed high levels of CD28, if we could block the B7, it's counter receptor CTLA4 interaction with the CTLA4 antibody, ipilimumab, then the B7 would have an unfettered direct activation of CD28, which was obviously a positive signal. On top of everything else, we thought CTLA4 would lower the threshold of activation of endogenous tumor reactive T cells. And since we weren't lymphodepleting these patients because we were giving them memory cells and we didn't need to lymphodeplete, um, by lowering the threshold of activation with ipilimumab, perhaps we could allow antigen spreading to occur. And unfortunately, that's exactly what we saw. In some patients, even those who have failed immune checkpoint therapy or failed ipilimumab, they received a single infusion of MART1 or GP100 specific T cells that were grown in the manner which I just described from their peripheral blood alone, no high dose lymphodepletion, no high dose IL-2, but followed immediately after infusion with ipilimumab, a CTLA-4 antibody, and you saw long-term persistent lead transfer T cells. You saw antigen spreading occurring, which we were able to measure in the peripheral blood, and you saw, in some cases, complete tumor eradication, bulky disease, even not just small volume disease. And so, spurred by that, in the Anderson, we started branching onto different trials. Now, one of the other major problems with solid tumor cell-based therapies, or even liquid tumors, is the dearth of TCR targets. And we saw that as a problem early on. And although there was NY-ESO-1 and now PRAME and a few other targets, got a bunch of CAR targets, the bulk of targets for a tumor really is a TCR target, because what the TCR target is the peptide MHC, and that peptide can come from any protein anywhere in the cell, whether it's transcription factor, an enzyme, or even an MDR gene, or DNA repair, and so on, cancer testis antigens. And Capon and Yulin Chu in the lab, they actually went about starting from ground zero, literally just ignoring what was already being used in mass spec, and built a pipeline for antigen discovery, running about 20, 30 lines, 80 runs, 500,000 spectra, with a specialized mass spec algorithm that were now eluting off about, I guess they had about maybe 6,500 peptides of high confidence that would elute off MHC. So these weren't predicted, weren't where the money is, elute off the MHC. And then we passed that through a number of filters to be sure that they were more tumor selective. Now remember, this came from the MHC that these tumors are presenting. So it wasn't a prediction. We just knew it was processed and presented. But then on top of everything else, we took the ETC workflow that we use to generate T cells. And we knew this ETC workflow was robust because when you do a clonal type tracking analysis, we knew that the T cells that we had isolated from patients specific for a particular antigen, when we look at that TCR clonal type, we go back and do PCR on those patients before infusion. We cannot find it. You know how sensitive PCR is, 1 in 100,000, 1 in a million. So we knew that this method of ETC workflow was sensitive enough, or robust enough to pick up T cells that were less than 1 in a million in frequency in the peripheral blood. And so we used this to validate those peptides, which we had eluted off the tumor HC, so that we could grow the T cells and then prove, in fact, empirically, in vitro, that the T cells that were specific for that candidate epitope could now recognize and kill a tumor endogenously expressing the MHC restriction, and also endogenously expressing that antigen of interest. So kind of the whole gamut. The tumor antigen was being processed and presented a peptide of sufficient MHC density on the surface to elicit a T cell response, and that T cell receptor. That T cell response was a sufficient affinity to recognize the peptide we see on the tumor, and then affect the killing and release of enzymes and so on. So furthermore, we took the TCR, sometimes sequenced that, and would engineer and put that into just lymphocytes and direct specificity. So now we had this pretty extensive portfolio, and when you look at it, we were shocked to find actually that source of some of these peptides was not just CT antigens, for which we had many, and not just ABCB5, for example, and MDR gene, and not just RAD51, and not just DNA repair genes, but actually cell cycle proteins and chemical complexes. And who would have thought this was immunogenic? And so then we started working with some people in the rare tumor field, Fred Lang and Stephen Lai and Bernard Ravi and Tony Conley, to look at glioblastoma, anaplastic thyroid cancer, angiosarcoma, and chordoma, respectively, if I match those names and diseases right, and show that as long as that rare tumor expressed the antigen of interest that we could find in our immunopeptidome portfolio, we grew T cells, so those targets, they killed the rare tumors, and probably something that's not been shown before. But I think in the midst of all this, we're also studying how memory occurs, how to prevent exhaustion, together with Kunal Rai and a few people in the lab are working on this rigorously to see if we can knock out certain genes or certain epigenetic higher order programming in order to make the cells resist to exhaustion. We have an exhaustion model. It's all in vitro. This is all human T cells. And I think ultimately our goal is to, bring adoptive cell therapy not just to patients selective, either because of the toxicities, a highly selective population of patients, but to all patients. And not just obviously patients who are HLA-A2 positive, but Asian patients and patients who have different HLA alleles, not just A2, A24, A3, 11, and so on, but also to understand the mechanism of resistance and to engineer the cells in a way, preserving their normal T cell receptor circuitry, just nudging it in the right direction to give it the advantage it requires to undermine tumor immune evasion. I'm going to stop there. Thanks very much. I think I've spoken my 5 minutes. Just remember, you can't make this stuff up. You have to prove it. There. Okay. All right. And come to the lab with a clean slate, naive thinking, and some of the best questions are the ones that aren't asked. Thanks very much for your time. And for those who don't know how they got here from YouTube, you probably got here through the website. And let me just put that up here so you can poke around what else is going on in the lab. There we are. There's a QR code for it. And somewhere down here, there we are, is the website, www.tsellsrus.com. Hope to see you around. Take care. Bye.